Okay, so a brief summary of the webinar before we open up for your questions. So the use of IPD in meta-analysis is increasing. It's becoming a very popular approach because it has many advantages. It allows a very flexible and complex analysis approach, and it truly is the only option for some clinical questions, such as the epilepsy reviews that I've highlighted here today. But IPD meta-analysis is really hard work, and this sort of approach should not be entered into lightly. Requesting and preparing data is very time consuming. As I've highlighted, some of these projects have taken over 7 years to complete. And the interpretation of the results is directly dependent on the available data, so availability bias should always be kept in mind when interpreting these results. Also, this sort of work, while it is considered to be gold standard, it does require significant statistical expertise, and this sort of work is not automatically free from bias. So I do endorse the use of the new PRISMA IPD checklist to ensure high-level conduct and reporting of IPD meta-analysis. And the final thing to bear in mind that although IPD has a lot of advantages, maybe gold standard, it may not necessarily be the best answer for every question, and actually an aggregate data meta-analysis may be able to provide a mathematically equivalent answer in a much quicker amount of time. So the first thing that should always be considered when considering an IPD meta-analysis is whether IPD is required or not. So to IPD or to not IPD should always be the first question. I've got some references. These slides will be available afterwards, Methodological, and also the Cochrane Epilepsy IPD reviews. And thank you very much to everyone who's involved. This was definitely group work. And now I'm very happy to take your questions. Thank you.
Wonderful. Thank you so much, Dr. Nevitt. This was a very, very informative presentation. There are a couple of questions that the people here, the attendees, have raised. First of all, several people asked if we're going to share the slides. So as Dario said, we will be sharing the slides and the recording of this webinar. And there are also a couple of questions that have been raised during the presentation, and please feel free for the remaining attendees to raise your questions in the questions panel. So one question that was raised by Mohammed, it says, it is regarding your network plot. So he says, does the number of node size represent the number of studies or the number of participants?
So in the network plot, the number of nodes, so there are actually 10 nodes, as there are 10 different drugs, and the size of the nodes, so how big they are, represents how many studies each drug has been included in. So the bigger the node, the more studies the drug has been included in. And actually the lines in between the nodes represent the number of patients that have been included in a pairwise comparison between drugs. So a thick line indicates that a lot of patients have been included in that pairwise comparison, whereas a thinner line shows that it's less patients who've been included in that head-to-head comparison. And if there is no line in between two nodes, then there hasn't been a study conducted comparing those two treatments.
Great. So the next question is by Diana. At the end of the webinar, it would be interesting to understand more about CSDR. So if you can provide more details about CSDR.
Well, actually, if you visit the website itself, it's got some very, very useful user guides and step-by-step sort of information and processes of how you go through and request data, whether it's the right place to find the information that is relevant to you. So actually, I would recommend just visiting the website. I'm sure that will give a much more full description of what CSDR can offer to Cochrane users better than I can.
Thank you, Sarah. So the next question is from Frank, who says, what Stata command do you use for IPD and MA?
So I was using a mixture of MV meta, which is actually multivariate meta regression, which has some some applications for network meta-analysis, and also there's a suite of packages that was produced by Professor Ian White called Network, which are very useful for a lot of these sort of commands. So do have a look at those.
Okay. The next question says, is IPD good to use for network meta-analysis of non-pharmacological interventions?
I'm not sure I can give a yes or no answer to that without knowing the sort of context of the clinical question. I would suggest thinking about some of the earlier questions that I posed about whether it's a complex approach that would be required for the objective, and thinking about the outcomes that would be involved, and whether IPD could be useful in that case. I don't think I could automatically say yes or no to that.
Thank you. So Sabrina also asks, which program is commonly used for statistical analysis? Data? Are analyses like multi-level analysis with study as a clustering variable, or does it require a specific technique and package?
Well, I personally use Stata as a package, and Stata has a lot of network meta-analysis commands. It also has commands for IPD meta-analysis itself. There is, I think it's called IPD metan, the command in Stata. I don't know of these specific commands. There's also a lot of commands within R to do meta-analysis and network meta-analysis, and in a lot of cases, depending on what the approach is that you're using, whether you're using what's called a one-stage approach of analysing the data to get a pooled result, or a two-stage approach, so analysing the data to get an effect size and then combining the data, then you may be able to use any package that you're used to, and then combine data within Review Manager, depending on exactly what it is that you want to do. So I would suggest using the appropriate software for the actual statistical technique that you want to use.
Perfect. So then Susie asks, in your epilepsy setting, you did IPD as primary analysis and IPD plus aggregated data as sensitivity analysis. Do you think that's always best, or could do IPD plus aggregated data as primary and then IPD only as sensitivity analysis?
So in the specific case of the epilepsy work, as we had concerns about the definitions of the outcomes, and also about, in some cases, the quality of the information we were extracting from the papers themselves, the aggregate data, we thought it would be better to do a sensitivity analysis. I would only recommend doing it, IPD plus aggregate data, as your primary analysis if you're very confident about the definitions of your outcomes, about the quality of the aggregate data, that it is of equal quality, or certainly of comparative quality to the IPD that you looked at yourself and perhaps standardized. But otherwise, if there are any concerns that the aggregate data may be slightly different to the individual patient data, then I would recommend a sensitivity analysis.
Great. So next question says, thank you for this very excellent work. Do you think IPD meta-analysis would take a shorter period in the future than it took to your team? Do you think it is expected from journals, ICMJE, or trial authors? What do you think?
It's hard to know for every project how long it should take, because this particular project had delays in many different areas. It would be hard to know now whether requesting information via data sharing platforms, if we've been doing that from the start, would have made things quicker, or if there would have been other issues. So it's really hard to say, but I do hope it wouldn't take now 7 years to do a project like this, as this project was being done through a massive period of change in this research field. I would hope the time would be reduced. And in terms of what journals expect, I would hope that journals would expect to see this sort of work reported very well, so reported using perhaps a PRISMA IPD checklist to avoid maybe some of the problems. For example, I highlighted that quite a lot of these IPD meta-analyses don't acknowledge availability bias, so that's something which is in the PRISMA IPD checklist, that it should be acknowledged if data isn't available. and what that could mean for the results. So hopefully we'll see a lot more journals encouraging use of that checklist.
Wonderful. So there are several questions. I will probably say a couple more, and then we will probably close this webinar because we are already ahead of time. The next question says about RevMan. If RevMan actually is a suitable software that can be used for IPD meta-analysis?
So directly, no, but it depends on the exact technique that you are using. So if you're using what is called a two-stage approach, where you are analysing the IPD to produce an effect size, so for example, an odds ratio or a hazard ratio, and then you are combining the data that you've used the IPD to estimate that effect size, then you can use RevMan. But if you are using techniques such as treatment covariance interactions, any form of modelling, then no, you would have to use a statistical package and then summarise your results within RevMan. You can import different figures, different plots into RevMan. So that's what we did for our reviews. We haven't used the data analysis forest plots in RevMan directly, but we've imported in plots that we've produced in statistical software.
Great. And the next question says, thank you for the wonderful clear talk. Is the commitment to sharing IPD usually made at the protocol stage by your trialists?
It would seem now, in most recent years, that is something that is being required by a lot of journals, such as the BMJ journals now require a data sharing statement from any teams that are publishing their protocols. So it does seem to be encouraging with data transparency policies, but whether this means every single team will always share their data is maybe a slightly different question, but at least there's certainly now a lot more awareness of sharing of data for projects such as this one. But I think it's going to take a few years to see whether everyone is actually sharing data who says that they are willing to do it.